And with that, we move on to our next speaker, who is Anne-Sophie from Institut Curie, and she's part of our France-BioImaging Node. So Anne-Sophie, please go ahead and
Yes.
start with your presentation.
Thank you very much. So, hello everybody. My name is Anne-Sophie Mace, and I am working as a bioimage analyst at Institut Curie. But I'm also part of France-BioImaging, and it's more that I will present today. I will present a project that we are leading with Jean-Yves Tinvez within Institut Pasteur, which is the hub of the analysts of France-BioImaging. And our main goal ultimately is to create an operating core facility for image analysis that would be nationwide and remote. So thank you for the invitation. We are happy to present that today. So Dorian presented a bit France-BioImaging, so maybe I will not go into details, but what I want to say is that today, even if you have the most beautiful images on any microscope, you will really need for publication to have quantification. And this is something that, is really known. And in France, there is really we know that, and we know that there is a need in image analysis, but not only tools, really analysts that will help using those tools or building workflows from those tools to really answer a customized question. So recruitment is happening, but most of the time, for the moment in France, people are hired in like microscopy team or in a research team, meaning that they are quite isolated. And the idea of France-BioImaging was to offer, sort of a community for those people, because there are some risks in the fact to be isolated. Is that we can sometimes a lack of local critical mass, and we really don't have people in our environment with the same skills and talking the same language, and we are more focused on the end application for the biologists instead of also our technical skills. So the idea of the hub of France-BioImaging, FBIAS, was really to create a community that includes all the willing analysts of France-BioImaging that works in a core facility and to meet regularly. So now we've decided monthly, and it's a remote meeting. And really, the idea is to help each other and to improve technically and scientifically. It's really to have a moment where we can talk image analysis with people understanding, to get some advices, and also to present some tools we developed. So this is something we've started in 2021. And this was the first phase of this project, and it's going on. So here it's the current team of France-BioImaging hub, and it's increasing. Day by day, maybe not, but year by year, depending also on the new French nodes that are created or joining France-BioImaging. And so from that, we really, us as analysts, we really. It really helps us, this group, it helps us to talk scientific image analysis. So the next phase was, okay, what can we do, this group, to help the FBI community, the user of France-BioImaging? And for that, we divided that in two phases. So the first phase was what we called open desk, that is opening free consultation for all users in France-BioImaging. So this is like the user, they receive an email by Caroline Thirier, who helps us a lot. And then they can book a 1 hour slot during which 2 or 3 analysts of France-BioImaging will help them on a specific question. This is free for them, and discussions are private and confidential. And this is really active meetings, meaning that people come with a question and we really try to help them so that at the end they have a tool or a new idea of how to do their analysis. But this is really a 1 hour slot, meaning that nothing happened before or after. just we ask for having the data before so that downloading them is not a main problem during the meeting. So this is something that we started in 2022, and that goes quite regularly every 2 or 3 months, depending also on abilities of people. But of course, we know that 1 hour is not always sufficient for those problematic. So here comes the following step, which is collaborative projects. So for collaborative project, we decided to make a proof of concept in the end of 2022. This was following an open desk where we saw a project that could lead to a tool that would be extensively used by the user who is in Montpellier. And this was done by an analyst in Nantes who was mentored, if necessary, by somebody in Strasbourg. And this is a tool that people are currently using and quite a lot. So we were very happy with this first proof of concept. And so this is something we want to continue in 2024. And this is something we can do because France-BioImaging founded 2 temporary contracts, whose mission are not only that, but part of participating to this collaborative project. So of course, we are building a bit the project. So what we want to offer is either the development of a specific tool with a training of the user, or maybe the quantification itself in case of tools that cannot be really transferred, or maybe end of paper. And of course, this is projects that are built because the idea is really to make a business plan also. We are still thinking about how we can organize these things. So really, we think that mentoring, asking junior analysts to do them with mentoring is the best idea. And we are already trying, starting some projects. So this is really what is in progress right now. And a few words to say that if you are asking for image analysis project on Euro-BioImaging, you could in fact be put in contact with us. So either directly by choosing the French node, or by asking for any node, maybe the message will come back to us. So we may work together. So here is a bit the context, but I wanted also to present you one project that was done so that you have an idea of what we can offer. So a concrete example. So I am an image analyst, so the biology will not be super precise, but anyway, I'm focusing really on the image analysis. And if you have other questions, so I will tell at the end, there is a paper that is in publication, so do not hesitate. So I will present a project that was done in Institut Curie with Alexandre Baffet's team, who is working on organoid and cerebral organoid. So the idea was to make some movies, so some time lapse in 20x on a cerebral organoid, just so that you have an idea of the size. So one organoid is more than 1 millimeter, and then one movie is really a small part of the organoid. And then later, after all these movies, the sample is fixed and imaged with immunofluorescence. And then we have new images, and the idea is to recognize cell from live imaging. So the last time point should look the same as the fixed image. So what kind of tool did we develop for that? So we decided that we would work on the contour of the full organoid. So we would segment organoid in the live sequence and in the fixed sequence, and try to align the 2 contours. So here, for example, we see that we need a flip and a rotation of 33 degrees. And applying this transformation to the last image, we can refine the corresponding cells. Okay, so now how do we do that? Because that's why I showed you what was the 20x. It's a very small part of the organoid. So with this, we cannot have the full outline of the organoid. So to do that, we had to adapt a bit the acquisition and to add at the end of the acquisition an image in transmitted light modality, at the same position, but in 4x, to be able to obtain an image where we have the full outline of the organoid. For the fixed sample, it's easier because it's fixed, there are less constraints, so we went for a mosaic in 10x. And so here is an example. So here are some movies, so the last time point of the movies and where they were on the organoid. So here is a fixed sample that was obtained. So how does this work? We look for contours in both images. We try to align them automatically, to find if there was a flip or not and the best rotation to align the contours. And when it's done, we apply that on the fixed image, and we can really see both images. And if I want to know what was going on in the movie that was taken here, I know it was taken here in the sample. Because the 10x didn't give enough information, then the user goes back to the microscope to analyze here on 40x. And then, because maybe I should have said that. So the purpose of all of that was to understand a bit how cells were dividing and what they were becoming, and if this division was symmetrical or not. So here is a small example of a cell dividing, and the idea is to know, okay, what is it becoming? So if we look at the last image of the movie, we can find those two cells. And then after going back to the microscope at the 40x, we can see those two cells. And with the different markers that were imaged, we can see that those two cells are intermediate progenitors, meaning that this was a symmetrical division. And then this study was done for a lot of cells to obtain statistics about the division of the cells in those organoids. A few words on how it was done. So it was, in fact, a Fiji macro first that was doing all the work construction and segmentation, and then MATLAB code with, in fact, two interfaces, because I didn't say it here, but most of the time there was not only one organoid acquired, but several. So we were segmented several of them in live and in fixed. So we had to associate them and then align them. So here is the second interface for alignment. So there was an automatic alignment for, let's say, easy segmented organoid. But of course, sometimes, because this happened in biology, it was not perfect. So here is an example of a broken organoid. So we also wanted that to be analyzed with the program. So there was all a manual transformation where you could manually align some part of the organoid, seeing that they were corresponding. And this is, if you are interested, the paper is already on bioRxiv, and hopefully it will be available in Nature Cell Biology quite soon. And once again, this is a collaboration with the team of Alexandre Baffet, with Laure Coquan and Clarisse Brunet especially. A few words to say also thank you to all the analysts of France-BioImaging, because this is working because they are involved. Thank you for the invitation for this presentation. I'm very happy. Thank you for the team that helped me on this project. And thank you to all of you for your attention. If you have any questions.